Hi, everybody. We're group number one. My name is Zachary Hoskins, and my partners here are Mason Hoffman and Jack Damewood. Our presentation is titled Mitosis and Cancer Research Treatments that Arrest Mitosis in Targeted Cancer Cells. Our question, as we began to think about cancer and mitosis, was, can treatment that arrests cell mitosis be used to target and treat cancer cells? From this question, we hypothesized that using antibiotics to slow or stop the cell cycle will decrease the rate of cancer growth and increase the body's immune response. Our prediction was that in cancer treatment trials, the more success in slowing or stopping cell division in cancer cells, the higher the rate of remission or mortality rate in patients.
All right. So this goes back to our question, can we use antibiotics to stop cancer cell growth? Cancer is a disease in which cells divide uncontrollably. Cancerous cells arise due to genetic damage of the cells. This damage results in the cell cycle and division of cells not being regulated correctly. This is usually due to the cell not undergoing apoptosis or programmed cell death. Cancer can lead to damage of body tissue and in some cases can lead to the death of the host of those cancerous cells. Many scientists have researched ways in which these cancerous cells can be controlled. Recently, this research has included the study of antimitotic chemotherapeutic agents to promote apoptosis of cells that are dividing uncontrollably. These agents are further being researched to provide more information on how they induce apoptosis on the molecular level. The goal of our research in this group is to further analyze how antimitosis agents can reduce the uncontrollable growth of cancerous cells. Our research will further investigate how these agents can target cancerous cells and promote cell death to prevent the formation of cancerous cells and tumors. So, our early findings from our question, if antibiotics can stop cancerous cell growth. Our early findings showed that treatments which targeted microtubule formation, such as taxanes, are effective in treatments against some solid tumor cancers, but with significant side effects. Preclinical models show promise with inhibitors designed to target PLK1, AURA, and B and EG5, CNPE, those cellular signals. Testing of these inhibitors in a clinical setting, however, did not produce the desired result of cancerous cell death. It has been concluded that these inhibitors must be paired with other treatments with improved pharmacodynamics to prolong retention of these inhibitors in tumors. Experimental analysis showed that diazoxide induced caspase 8 apoptosis. This resulted in the failure of the MAD2 spindle development checkpoint. The breakdown of the meiotic spindle was shown to mainly induce apoptotic cell death rather than necrosis or senescence. Taxol was also shown to induce MAD2 checkpoint failure with a 58% cellular survival rate, whereas diazoxide was shown to induce MAD2 checkpoint failure with a far higher cellular survival rate. And we can see that here, represented on this graph, with Taxol treatments being represented in blue and diazoxide treatments being represented in orange. As shown in the figure, a Taxol and diazoxide treatment demonstrated similar results in the killing of cancerous cells. If you notice in the figure, after a concentration of about three times concentration, both antibiotics' effectiveness in killing cancerous cells level out rapidly, leading us to infer that increasing concentration past 10 to the power of 1 does not increase the antibiotics' effectivity. And in this figure, we can see a basic breakdown of the antibiotic agents and their functions. One antibiotic agent function is to target cellular inhibitors, as seen on the right. These inhibitors are basically checkpoints that stop the cell from proceeding in the cellular cycle, such as AAURA, AAURB, EG5. Whereas on the left, some antibiotics such induce cellular death, like apoptosis or necrosis. Diazoxamide is one of these that produces cellular death.
So kind of getting into our methods. Thanks, Jack. So one article we found that was titled Antimitotic Therapies in Cancer examined the use of the specialized inhibitors that we talked about that target PLK1, AURA, NB, EG5, and CENPE, and showed that they were effective in preclinical models, however, fell short in the clinical setting. This led to the conclusion that a need for additional treatments in addition to antimitotic therapies to target and arrest mitosis in cancerous cells. So another article titled Antimitotic Chemotherapeutics Promote Apoptosis through TL1A Activated Death Receptor 3 in Cancer Cells studies how chemotherapeutics promote programmed cell death of cancerous cells by disruption of the mitotic spindles. This article studied diazoxamide, which we talked about, and how it inhibits cell growth by disrupting mitosis under that apoptosis in the summary. The antimitotic diazoxamide was compared to Taxol, which is also an antimitotic. And then diazoxamide was further tested to identify if cell death resulted in apoptosis or necrosis by testing HELA cells with different doses of diazoxamide. So moving into our discussion, the experimental results concluded that while the antibiotics were successful in lab settings, the clinical testing results proved far less effective than originally postulated. Why was this? One theory kind of suggested that T cells are just as susceptible to the antibiotics as the cancer cells. And so because these T cells play a big role in immune response and tumor suppression, the treatments that target both of these, the cancer cells and the T cells, have a pretty large effect on the immune system and are less effective in a clinical setting. And another possible explanation for the poor results of the clinical studies is the unknown amount of meiotic cells in a tumor's area. So tumors that contain fewer meiotic cells than previously imagined, because of this, the antibiotics did not have a sufficient substrate to produce an effective result in the clinical trials. One idea to circumvent this problem suggests that maintaining concentration of the antibiotics in a tumor over time might increase the effectiveness of the treatment. If the antibiotics are present for a long period, the more meiotic tumor cells they have, the greater chance to promote apoptosis in the cells. So overall, the antibiotic treatment has shown promise with their ability to target cancer cells and promote apoptosis. Further experimentation is needed to conclude whether these treatments can be altered to target primarily tumor cells while not affecting T cells, which was kind of illustrated above. Always more research. These are our references. Thank you for listening. This was super interesting for us to kind of learn about. You kind of hear, and obviously cancer is super prevalent nowadays, and trying to fight it is something that there are new studies coming out every day. So we found that this was a study that could really branch off into some different directions. So these are the references. This is where we found a lot of our material. We're super looking forward to kind of diving into this more, and thank you for listening. Thanks, Jack. Thanks, Zach.
Thank you.
Take care.